It's a pleasure, we can see if we can do this, to now introduce our first speaker and get us going. You've always been big in my world, so really great. So it's a pleasure to introduce Dr. Lila Davachi, who's a professor in the Department of Psychology at Columbia University. Lila did her PhD at Yale, working with the late Pat Goldman-Rakesh, and then a postdoc at MIT. Before joining the faculty at Columbia, Lila was on the faculty in the Department of Psychology at NYU from 2004 to 2017, and at NYU she also was the director of the Center for Learning, Memory, and Emotion. She's a leader in using behavioral and neuroimaging investigations to characterize how humans encode and consolidate their experiences in memory and how memories are organized into knowledge. Among her many contributions, she has conducted seminal work on the differential roles of medial temporal lobe cortical regions and the hippocampus in building memories. And as we'll hear today, she's also advanced understanding of how memory, how memory replay supports consolidation, as well as how encoding and remembering are influenced by states of neural systems. Her work's been recognized with numerous awards and honors. Earlier in her career, she received the Young Investigator Award from the Cognitive Neuroscience Society. More recently, she's the recipient of the Lenfest Distinguished Faculty Award and an award for cultivating science identity and increasing persistence in STEM. She's an elected fellow of the APS and an elected member of the Society for Experimental Psychologists. Lila, unfortunately, is why she's 7 feet tall rather than 5'7", 5'8" feet tall, if I'm maybe about my height. Unfortunately fell ill earlier this week. There's a bug going around the East Coast like there is around here, and she wasn't able to travel out and join us, but we're lucky to still have her here via Zoom. Today, her talk is going to be about the life of a memory, post-encoding reactivation, and reorganization of episodic memory. Please join me in welcoming Lila.
Thank you. It's really incredibly fascinating to be able to still be there, even though I'm not there, but I'm very sad not to be there with you. Just a note that I may end up coughing, and I have a lozenge in my mouth, so I may mute and sort of take the talk a little bit slowly. I apologize if that ends up happening and I'm no mute in time. Okay, let me share my screen and hope that you can all see that. And are you able to see my cursor?
It looks good, Lila, and we can see your cursor.
Great. Okay. I've been working on the study of episodic memory for over 20 years. First and briefly in the rodent as an undergraduate, and then in monkeys for my graduate work, as Anthony mentioned, and in humans ever since. And in all of that work, the major goal of mine, and I think probably one that's shared with many of you there in the audience, was to understand how memory works. It seems like a rather simple question. However, the very question assumes that we have an understanding of what counts as a good memory. It assumes that memory is for remembering, full stop. If it was that simple, if really that was the answer, if it was that simple, then evolution has done a terrible job, since most of what happens to our memories is forgetting, even in the natural state. So instead, if we formulate the question a little differently and ask, what is memory for? We may quickly rest on the idea that it is for crafting and updating our knowledge to best represent what we might need in certain contexts to promote adaptive behavior. So now, if we start from this question, what is memory for? The answers that we get to our first question may differ drastically based on so many variables that we've been thinking about that define our experience. One of the challenges of studying memory is that there are so many time points during which various factors are known to influence whether and how an experience is remembered. It's a highly multidimensional problem. Take the time of encoding. At the time of an experience, or rather, encoding, experiences are associated with a dynamic pattern of activation in the brain. And I'll just put up a pretty little engram here. So this is supposed to represent a dynamic pattern of activation. At retrieval, when presented with a partial cue, successful memory retrieval has been hypothesized to be associated with the reinstatement of patterns of brain activity that were present during encoding. And much work now has shown that the extent of overlap between encoding and retrieval patterns in the brain relate not only to successful memory retrieval, but also to how many details are remembered, as well as how confident one is in your recollection. Now, these are both what I would call online states, and there's a lot of work that's been done to understand the mechanisms that are at play during encoding and retrieval. Now, there are also offline states. During offline states, in between encoding and retrieval, it is known that patterns of activity associated with the experience replay or reactivate. And we're just beginning to understand the functional changes that replay promotes. One of them, of course, is increased synaptic plasticity in hippocampal neural ensembles recruited during encoding. So this post-encoding replay is a leading mechanism that might support not only the strengthening of relevant aspects of those experiences, but perhaps also their reorganization. I probably don't need to spend a lot of time introducing the idea of hippocampal replay to this audience. Hippocampal replay was first identified in the 80s, and it primarily was shown during sleep. Here's an example of place cells that are activated while an animal is running on a maze. And what was first discovered, it really changed, I think, the history of memory research, is when the animals went to sleep, you could see that those same place cells reactivated in temporal order, and also importantly at a compressed time scale during sleep. Another way to look at this data is to look at the correlation between pairs of CA1 neurons while animals are running on a maze and their pre-slow wave sleep periods before that experience, and see that that correlation structure, the correlations between these neurons, increases after an experience. And I show the data in this way because it's very similar to some of the ways that we've analyzed our data. And critically, there is some work that has shown that selective suppression of hippocampal replay during offline periods can impair spatial memory. However, while much of the animal work on replay focuses necessarily on local replay in the hippocampal system, we hypothesize that the ultimate determinant of aspects of new experiences, or what aspects of new experiences will become long-term memories, should involve systems-level hippocampal cortical replay. So this coordinated replay has been shown during sleep in a handful of papers in rodents. Importantly, in 2012, Logothetis published a study with monkeys that were in a scanner while also recording from hippocampus. And in that study, they were able to identify moments where sharp wave ripples, which are the high frequency events where replay is often seen. So coincident with those sharp wave ripples, that's the 0 point here, he was able to show that there are increases in bold signal in the primate in cortical regions. There are also, interestingly, decreases in other subcortical regions. So that the idea here is that the ripples themselves are triggering an increase in activity in cortex. And in humans, a recent paper showed a similar kind of result. This is from Norman et al. This is now using ECOG in human patients. And these are also triggered on ripples or peri-ripple high-frequency activity that's been identified in the cortex. And the larger, redder circles are where a lot of that high-frequency activity is seen coincident with sharp wave ripples in the hippocampus. So clearly, these ripples aren't happening in the hippocampus in a siloed fashion, but they are triggering activity in cortical networks. So for the purposes of today, I'm not going to focus on only one cortical region. I'm just going to refer to the cortex because I want to take a bird's eye view of hippocampal cortical interactions, as they do seem to be global. But there are clearly also important and interesting particular cortical networks that appear to be critical in memory replay and stabilization. And I will mention the regions in each of the experiments I present, but I'm happy to take questions if you want more detail. And so today, what I hope to go over is put forth the idea that the most diagnostic biomarker of long-term memory and stable memory is a distributed representation of an event across hippocampal cortical networks. I'll talk about our work on offline replay and ask, what does it represent? And does post-encoding replay, how does it reflect the strength of prior memories? And then near the end of the talk, I'm going to talk about reorganization. So during these offline states, it looks like memories are reorganized according to their regularities. And I'll tell you about a couple of experiments if we have time, but I'm notorious for trying to fit in too much detail. So I apologize if we don't get to all that data. Okay, for this first question, just to take a methodological moment to say there are many ways that we have looked at offline replay, but they all share a similar structure. So we give individuals an experience, we call this the encoding period, where they're exposed to many, many trials during encoding. And then we can identify patterns of activity that are local activation patterns. We've looked at multivoxel correlation structure as well as systems level functional connectivity that characterize the encoding experience. And then we look for the presence of those patterns during post-encoding rest periods. And critically, all of these studies also include a baseline period. So we can ask specifically whether there's evidence for experience-dependent increases in the similarity or replay of those encoding experiences into post-encoding rest above and beyond what might be there during baseline rest. So in our first paper that was published in 2020, Ariel Tambini actually showed for the first time that you can examine rest periods in humans and see evidence for memory replay. And here's just a snippet of the design. Again, people had a baseline rest scan, and then they were exposed to object-based trials in an associative encoding task, and then a post-object-based rest scan. And the main surprising effect from that experiment was that if we look at connectivity, this is the y-axis, it's just correlated activity between the hippocampus and a visual cortical region, LOC, both during the baseline rest and the post-task rest, we saw a significant increase in connectivity that lasted the full 8.5 minutes after this object-based task. Participants also did a scene-based task, but I'm not going to focus on that today. But critically, what linked this experience-dependent plasticity with memory? What we showed is that in this post-task rest period, the level of connectivity and actually connectivity increase from the baseline rest to the post-task rest across participants predicted how many of those object-based trials they would remember later on. So memories on the x-axis, connectivity on the y. So there was a strong correlation between post-encoding connectivity and memory for the preceding experiences. Now, in this next study, Kaya went on to ask, well, what about if we activate memories after a long delay versus a short delay, knowing now that what happens immediately after learning is this communication between the hippocampus and the cortex. So in this study, we had people in the scanner while they were reactivating 24-hour memory, the long delay, or a short delay memories that had just been encoded right prior to the reactivation. And then we tested memory on across two tests to allow us to look for forgetting over time, which is a critical behavioral signature of memory stabilization. Again, these were novel object associations that people encoded. And here we focused on perirhinal cortex in this study. And what you can see here is that hippocampal perirhinal connectivity is significantly increased for items that are reactivated after 24 hours compared to items that are reactivated after only a few minutes. And this was, again, domain specific, which is another interesting component of these post-encoding correlations. Okay, so again, we see an increase in connectivity, but is that related to their stabilization? Is this related to the extent to which these memories have been consolidated? And now I'm showing you brain behavior correlations. So on the y-axis is the connectivity measures across participants, in this case, hippocampal perirhinal connectivity. And on the x-axis is the amount of forgetting that occurred for the specific trials from day 2 to day 3. And you could see that the greater the connectivity between hippocampus and perirhinal was correlated with reduced forgetting over time. So now it looks like connectivity is a signature of the strength of those memories, how much they're stabilized and how much they're resistant to forgetting. We don't see that same relationship for the memories activated after a short delay or for novel trials. So that was selective to the long delay trials. And a really important contribution of this paper is that Kai was then able to compare and ask whether overall activity in the hippocampus and also the perirhinal cortex, which is not shown here, predicted memory? And the answer was no. The overall level of activity during the reactivation had no relationship with subsequent forgetting for the long delay or the short delay trials. And interestingly, there was still a significant relationship for the novel trials. So novelty is best captured by overall activity. That predicts their forgetting rate. But once an item has been repeated and once it's been replayed for a period of time, it looks like connectivity is the best predictor of the longevity of those memories. So digging a little deeper, let's just review what we've shown across many papers, and I've only presented a little bit of the data today in the interest of time, is that immediate post-encoding increases in hippocampal cortical connectivity, I refer to as HCC here, are evident. They've also been shown to be related to long-term memory for preceding experiences. That's the data I showed you from Ariel's first paper, and then there are some subsequent follow-ups. Now, interestingly, increased hippocampal cortical connectivity has also been shown to increase when older compared to newer experiences are reactivated, and that's associated with more stable memories. And that's Kaya's data, but also a whole slew of other autobiographical memory studies have shown that same result. And data that I didn't show you today is that hippocampal cortical connectivity is also modulated by factors known to increase memory consolidation, such as reward or curiosity. But in this next experiment, which is an ongoing study that is about to be submitted, we wanted to ask whether memory replay, specific now memory replay of individual trials from learning underlie these connectivity measures. So, and specifically, we wanted to understand if offline replay reflects the prioritization of recent experiences. So what do I mean by that? In this study, Wangjing and Assia used a manipulation known to improve memory. Here we just chose simple repetition to see if replay differs across these conditions. And in particular, we wanted to look at coordinated replay and connectivity. So specifically, again, the design is very similar to what I showed you. There are encoding periods and post-encoding rest periods. In this experiment, we actually had 2 of those, and then we had a memory test. And the critical manipulation here was repetition. Some of the trials were repeated 3 times, and some of the trials were only presented once, so they were unique. And we know from decades of work that the thrice presented items will be better remembered, the associations will be better remembered than the once presented items. And indeed, that is what we see in the behavioral test. So the items that are presented 3 times show significantly greater memory performance at the end of the day compared to what we're calling the weak memories or the once presented items. Okay. So the analysis approach that we took here was now to define a vector of neural activity in each region of interest. And the regions of interest that we chose were the hippocampus, but also cortical regions that have all been implicated in memory representation and indeed in replay in prior studies. For each trial, we created a vector, and we were able to pass that vector across every TR of the rest period. And we could get a correlation value that represented the similarity between that trial and each TR during rest. And this is what the data would look like for one trial before thresholding. And then we applied a thresholding approach, which has been done in a couple of other studies, to pick out those trials that show the greatest similarity, and we counted those as a replay index. So after thresholding, we get an index of what trials are being replayed during what TRs during rest. And what we see, interestingly, is hippocampus. Hippocampus didn't show any difference in the replay index for the thrice presented items and the once presented items, even though memory was significantly improved for those trials. But look what we see in cortex. The ventral temporal cortex, retrosplenial cortex, posterior cingulate cortex, and MPFC all show significantly greater replay indices for the thrice presented items over the once presented items. Super significant, strong effect. More relevant to the talk today, what we see also is that coordinated replay is increased in these regions as well. And I'm highlighting the two that show the strongest effects, the retrosplenial cortex and medial PFC. These are both regions that have been implicated in the long-term storage of memories and in consolidation work that's been conducted in rodents. Coordinated replay here refers to the idea that can we see when replay is happening in the hippocampus at a particular TR, do we also see evidence that replay is coordinated, that it's also happening in a cortical region? So again, we see it looks like the repetition of items, the simple repetition of items is somehow accelerating the consolidation process and leading to increased coordinated replay in these immediate post-encoding rest periods. We then wanted to ask whether the replay indices here on the y-axis are related to the connectivity measures that I talked about earlier in the talk. And in fact, we see, this is fresh off the presses here, we see that there are correlations between post-encoding hippocampal connectivity and the frequency of coordinated replays between those regions. And so far, we're seeing that strongly in VTC, interestingly, for the once presented items, but with retrosplenial cortex now for the thrice presented items. So this actually helps us to feel confident that at least some of the time, our connectivity measures between regions are reflecting underlying coordinated replay, in addition to being able to actually measure the moments of replay that are occurring in the human brain. Okay, so the answer to this question, could memory replay of individual episodes from learning underlie these connectivity measures? And the answer there is a resounding yes. Another point I wanted to make is that Ariel Tambini went on in her postdoc and used TMS to stimulate lateral occipital complex, which was the region that showed hippocampal connectivity correlations with memory in her first resting state study. And stimulation to LOC not only led to a decrease in memory performance, so it interrupted the stability of those memories, it also disrupted the correlation between connectivity and memory. So this is highlighting the causal nature of the hippocampal connectivity and subsequent or ongoing consolidation of those representations. Okay, so now I want to turn to understanding now, what is memory for? This question of why do we gather all these memories? How does the memory system take in the information that we're experiencing, and to what end does it veridically represent those memories, or does it start to reorganize them based on our, let's say, our past experience? And we've begun to ask that question by presenting trials that contain regularities, much like in statistical learning paradigms might look for. So in this first study, we used, we asked whether memory integration, let's say behaviorally, but also in the brain, would occur on the basis of perceptual or semantic features of the stimuli. So in this experiment, people were shown unique trials that contain an object and a scene, but across the lot of trials, 128 trials, there were only 4 scenes. So the 4 scenes repeated. It was a beach scene, a jungle scene, a city scene, and a bedroom scene. All of the objects, however, were trial unique, and people were encoding each trial on its own. And what we wanted to ask is whether or not the brain comes to organize these trials according to their shared features. In this case, it was the shared scene, which is a perceptual stimulus, but also a semantic stimulus to individuals because they could label these, for example. And this is work done by Alexa Tampari when she was in my lab. And what we aimed to do was to look at whether the neural patterns that are evoked for each of these trials, whether in the end, what comes to be represented is the strong core, the regularity across those trials, of course, also with the details of the individual trials. This is just a schematic of our hypothesizing during the time. What we actually did is look at, let's focus here on the retrieval trials here. And I want to point out importantly that all of the imaging data I'm going to show you are from retrieval trials where the scene was no longer presented. And so people studied each object with a scene. And then during retrieval, and the retrieval happened either the same day or a week later, people now were asked to recover the association, the scene, only presented with the objects. And what we could do is ask whether the similarity between objects that had previously been paired with the same scene is now increased or more overlapping than objects that were paired with different scenes. In other words, this is our way of looking at what the brain is organizing experiences based on these shared features. And what we found, I'm just going to show you a little bit of the data from that study, is first I'm going to show you data from medial PFC. What's graphed here is inter-item retrieval similarity. So again, that's the similarity between distinct retrieval trials that shared the same scene in blue and ones that did not share the same scene in orange. At the immediate retrieval period, there was no evidence for an increase in memory integration. But what we saw at 1 week was that the MPFC neural structure reflected the similarities of those encoding trials. So now there was significantly greater overlap in memories that shared the same scene compared to ones that didn't. Now, importantly, we also saw this effect in the hippocampus. Again, there was no difference during recent retrieval, but at remote retrieval, we saw a significantly greater overlap in neural patterns for the scenes that shared features compared to the ones that didn't. So it looks like the hippocampus and MPFC are organizing experiences based on these regularities, and importantly, only at a delay. We don't see evidence for that at recent time points. So next we wanted to ask whether this neural overlap is related to behavioral memory integration. And to do that, we went to a different design. So Alexa, and this is about to be submitted, Alexa created a design where now the relationship between items was sequential and predictive. So in this case, there were multiple, let's call them triads or really quartets that were presented. So people would see A, B with 100% transition probability. And these are presented, I want to say about 20 to 30 times. So it's very much like a statistical learning paradigm. So people see A and B with 100% transition probability, and then B, the broccoli, is a decision point or a choice point. It either leads to what we call C1 or C2 objects. Critically, C1 objects and C2 objects are never presented in the same temporal context. In fact, they're by definition separated in time. So we wanted to ask, one, extending her prior work, whether C1 and C2 would become integrated because they're both correlated with the preceding items. They had the same antecedents. And then we also could measure behavioral integration by asking whether and how these two items prime each other, whether they're overlapping and they lead to, or they facilitate greater recovery of one when presented with the other. So we used an implicit recognition priming paradigm where we would present here, let's say we had FOIL items, baseline items, and then we'd have the C items presented back to back. And we would ask whether having presented one C item would lead to a faster response on the next C item, as if they now become integrated, like this picture, in memory. And what we see is in two separate experiments, at the remote time points, but not the recent time points, we see evidence for behavioral priming. So now with a temporal delay, these memories have become integrated such that they facilitate your responding to one of them when presented with the other. And that's presented here as a faster reaction time for the primed item compared to the control item. Now, looking at the neural data, again, at MPFC, we created integration models that looked for whether the patterns that are evident in MPFC look like C1 and C2 items have become integrated. So we compared the actual data to a theoretical integration model where there would be maximal overlap for overlapping pairs versus non-overlapping pairs. And what we see, interestingly and unexpectedly, is that MPFC is now separating out C1 and C2 representation as a main effect. So the MPFC is treating these as if they are different endpoints and saying, I think that's sort of an interesting question, is we can ask the memory system again, ask it what's important to you. But in terms of behavior, it still goes in the direction that we predicted. So the change in the change in fit to the integration model, post versus pre-learning. Sorry, I didn't go over the details here. So the more overlapping C1 and C2 items are in MPFC, the faster you are to respond to C2 items. So even though the main effect is one of separation, where there is overlap, and when it is there, that's when those items are priming each other. And finally, I have one final data point to present from this study that's currently under analysis, and this is data by Arlene Lormistoir. She was interested in now looking at the post-encoding rest period. So after learning these transition probabilities, we are able to now look for in a sequence that people are learning and ask, what does the memory system want to replay? Is it replaying the A items, which are the beginnings, the most predictive items in the sequence? Or is it replaying B items, which are predicted, highly predicted, but also are these transition points? In other words, you either are going to C1 or C2 here, so they may garner more attention and more relevance. And this is just really preliminary, but at hippocampus, what we are seeing, interestingly, is significantly greater replay of the predicted items B, which are also decision points for C. So greater replay of B items compared to A items, and also greater replay of C items compared to A items, which is kind of fascinating because A items are the ones that tell you exactly where you are. You know when you see an A that you're going to see a B. And I'm not, maybe when I say you know, I don't mean explicitly that people know this, but the brain is learning those patterns. But indeed, A items are somehow down-weighted in replay compared to B items and C items. And we're still thinking about this study, and data analysis are ongoing. So I focused a lot on MPFC, and I just want to highlight that there's some interesting work showing that damage to this region of the brain, in particular VMPFC, this is a paper by Spalding from Warren's group, using a design developed in Ali Preston's lab for measuring memory integration, shows that damage to BMPFC does not cause impairments in premise pair memory, so memories for associations that were directly encountered, but instead causes significant impairments in integrating across those memories. So when asked about an AC inference pair, this is where these patients seem to show significant impairments. So in conclusion, the distribution of a memory trace across hippocampal cortical networks is the single best predictor of memory stability, with a special emphasis on midline cortical regions, MPFC, and retrosplenial cortex. Memory transformations are likely becoming the rule for neural representations of memories, not the exception. And this is exciting time for empirical research because we're able to modulate the learning experiences of participants and then directly examine the features of these experiences that increase post-encoding replay. And we're studying the human brain. We're able to probe what are the behavioral consequences of the underlying neural representations with an extensive array of elegant assessments of behavior. And of course, given the hippocampal cortical connectivity, we think is now such an important biomarker, it's a prime target for manipulation. And there's been some beautiful work using TMS, both in the clinical realm as well as in healthy populations. And I want to thank those who did the hard work. The work of the folks I highlighted today are in red, and thank my funders and thank all of you. Should I stop my share? I just wanted to put this up too and just give a shout out to all the struggles that are happening in my home country right now.
Thank you, Lila. Thanks for that tour de force through the really impressive research from your team. We now have time for questions from the room as well as online. We can start with questions in the room.
Hey, Lila, this is Bill Newsom. I come at this, subject mostly from an appreciation of the animal work that has gone on, and a lot of what you've referenced, the replay events and shortwave ripples and that beautiful loga theta study, are happening on really short timescales, measured in tens or at the most hundreds of milliseconds. And the sort of patterns and replay that you've been demonstrating here take place across seconds or even minutes in some of your studies, if I'm not mistaken. And I'm wondering, can you just help someone like me relate the world that you live in with these studies with humans to the world in animal research? And I think Nicole Rust's talk may help us do that in the next talk, but I'm just interested in your take.
Yeah. Well, thanks for that question. It's such an important one. And we addressed this a little bit in our TICS paper, Ariel and I. One of the data points that's out there that I think can promote our thinking of integration between these methods is that when these shockwave ripples are measured, they obviously are very rapid, as you said, but the underlying, or I should say that the subsequent correlation structure that's induced in those neural networks can actually last for quite a long time. And they've been shown to produce increases in bold signal. So but critically understanding how and when shockwave ripples as the neural mechanism, what kinds of bold signal increases and pattern increases they do correlate with are going to be important. But they do produce longer lasting effects in the physiological data and in the bold data. So I think the best case scenario is that what we're measuring is the kind of the footprint of what happened rapidly, but leads to an increase in correlated activity for longer periods of time. But one of, you speak to my heart. We've just submitted 2 U19 grants to try to look at memory reactivation in the same behavioral paradigm in rodents and monkeys and humans. And sadly, it was not funded, but I hope one day we'll be able to do more direct comparisons and have a better answer to your question.
Thank you.
Jay? Hi, Lila. Thank you for the talk. I wanted to ask a time sort of related question too, but this is about a much longer sort of time span of consolidation. Going back to the literature on HM and studies from the Squire Lab and so on, consolidation is often thought of as happening over months to years and not just overnight. And I wonder whether your research has given you a chance to think about, the involvement of the hippocampus in these longer term things, or what happens eventually when something that I learned about 5 years ago gets to the point where, based on data from HM and other patients like that, we would think it's no longer necessarily hippocampal dependent.
Yeah, I think when we started doing this work, we purposely avoided using testing those questions that you're asking, because they are a different question. And what we have, so one of the things that's really exciting about this work is that if we think of replay, whether they be shockwave ripples or reactivation or hippocampal cortical coordinated replay, as the best mechanism we have to promote memory consolidation, it is happening right away. It is triggered, I think, in the moment. As soon as you walk out the door, actually, I think replay is occurring. And so consolidation processes definitely start immediately, if you think of that as a consolidation process. And I think they are probably going to be ongoing. But the question then remains, when does that initial consolidation process end? Is it 24 hours? Is it 2 days? We've looked out as far as 1 week to look at the representation of those memories in those last studies, but we haven't gone further than that in our prospective designs. People who have used retrospective designs, asking you to recall autobiographical memories, and that's more relevant to HM, and the work that you're referencing, do sort of suggest that perhaps maybe the recollection of years old memories no longer involves the hippocampus. That work is really hard to interpret, because the hippocampus is also going to be engaged and active in appreciating the details of those memories and perhaps coming up with new interpretations. So the idea of reconsolidation. So it's a really hard question, and I'm not sure it's actually a question that we need to start asking right now. I wasn't that hungry for the answer to whether it became hippocampal independent. What I think is important is understanding what the memory system is grabbing from our experiences, what it cares about, and how it's integrating it with knowledge. And I think we're seeing that happen right away. But the way to start doing that is to start to look at, like you said, longer and longer memories and understand what can happen. But I can't give you a good answer to that today.
Lila, this is really interesting. So the first two questions are about the time, and I wonder, I'm going to ask a question about the spatial distribution. So you emphasize that it's a distributed network within the cortex. And then the TMS in one area, the MPFC, seems to be shown that it's required. I wonder whether this distributed nature represent different parts of memories are stored in different places, or what about TMS studies in other parts of the cortices?
Yeah, so the TMS study that I highlighted was the disruptive TMS of Ariel was to visual cortex, and she targeted a region that showed the greatest connectivity memory relationship in our imaging study. So the distributed nature is so interesting. There's been a lot of work, both from the Norman paper, there's data there, as well as a new Nature Review Neuro paper from Battaglia that has proposed the cascaded memory systems model. But in that, he does a beautiful job of reviewing the extent of replay. Now, they weren't looking at coordinated replay, but the extent of replay in the cortex. And it looks like a lot of the action's happening in the midline cortical regions, the default, seemingly named default network, not very default, very active network. The Norman paper suggested and really did a nice job of, now I'm forgetting whether it's the Norman or another paper. Anyway, sorry. There has been work asking human participants to retrieve episodic memories as well as semantic memories. And while we didn't get differentiation across the midline being posterior cingulate, retrosplenial, and MPFC, there was no evidence in that paper for any kind of distinction about whether semantic memories are more anterior or perceptual memories are more posterior in that network. But what was evident is that that network was, when you were retrieving memories, there were sharp wave ripples in the midline system, and they were there whether or not those memories were semantic or episodic. And they tried to understand. So that's a beautiful paper trying to get at the question that you're asking. And this is something that we're actively doing right now. So I think the next few years of research are going to be looking at the systems that are, what specificity is there in these systems. But the midline region is one that I would put a lot of money on for being prima facie where our long-term memories are ultimately stored.
Time for one last question. Thank you.
Hi, Lila. This is Julie Hauer. Thank you for your talk and for especially the clarity for a cellular neuroscientist. It was very helpful. I understood most of it. I have two quick questions. One is, because I'm not as familiar with the data, are the points on each of your graphs an individual subject? And if so, do you have any interesting information about, person-to-person correlations that strike you guys? And, if you analyze them with like a paired approach, do you see anything interesting? And the second question is, I just returned from Europe last night where there was a time difference of 9 hours. And so this idea of consolidation and what occurs over the first however many days it is, and all the drugs that we often take to help with sleep. I wondered, I'm sure you have some interesting things to say about that. Thank you.
Okay, yeah. So to address the first, some of the graphs were individual subjects and some were individual trials of replay. But yeah, we can look at across subject differences. We haven't done a large enough study for me to really address that question. I think I would want to have 50 or 100 people to look at individual differences in this particular where we have a within subjects measure as well. But in the replay, the individual trial replay data that we have, one of the results I didn't share was that even though hippocampal replay didn't differ in terms of the frequency for the once presented and the 3 times presented items, hippocampal replay of the once presented items, the weaker memories, was correlated with recognition, retrieval memory that day. In other words, it looks like hippocampal replay could be rescuing those sort of once presented items more so than the strong items, which are now being replayed in the cortex. And then what we did is we looked at the people who aren't as good. So we broke up we had 30 participants into 15 who we called sort of poor performers and the 15 who were strong performers. And we showed that hippocampal replay actually benefited the poorer performers more than the strong performers as well. So that's the only bit of individual subject data I can share with you today, but it is really an important question moving forward for healthy participants as well as in clinical populations. In terms of sleep is the new smoking, I think. I think sleep is super critical. It's sort of the opposite, not the new smoking. You don't want to do it. But I think as we're all experiencing, sleep is going to be correlated with all kinds of health issues, cardiovascular, as well as cognition and mental health. And we've done only one sleep study in our lab, but the kinds of replay events and connectivity measures that I shared with you today are definitely ongoing during sleep. And one of the big questions in our field right now is how is awake replay differentiated from sleep replay? Are they quantitatively different or qualitatively different? And that's ongoing research as well. Thank you for your question.
Great. Thank you again, Layla, for really an outstanding talk.